Welcome everyone to this week's colloquium. I'm Hakan Turechi from Princeton University. It is my great pleasure to introduce the first speaker of the 2025 colloquium series, Professor Arvind Murugan. Arvind is an associate professor of physics at the University of Chicago and serves currently as a deputy director at the Center for Living Systems. I'd like to first share a brief bio. Arvind obtained a bachelor degree in mathematics from Caltech, earned his PhD in high energy physics from Princeton University, then transitioned to working on problems at the intersection of biology, theoretical computer science, and statistical physics at the Institute of Advanced Study, then the School of Engineering and Applied Sciences at Harvard University. Joined the physics faculty at the University of Chicago in 2015. Arvind's research seeks to uncover the fundamental principles that allow physical systems to learn, adapt, and self-organize through interactions with their environment. Drawing on ideas from physics, biology, and theoretical computer science, his work aims to reveal how intelligent behavior, such as error correction and associative memory, arise naturally from dynamics of biochemical networks, self-assembling materials, mechanical systems. Arvind has a very interesting background that winds through string theory, statistical physics, and biophysics, giving him a unique lens through which he is able to explain and demystify complex concepts. I've always found this perspective deeply insightful, and I'm confident you will as well. Right. So please join me in welcoming Professor Arvind Murugan.
Thanks for that kind introduction, Hakan. Much of it exaggerated, but one thing that's true is it's a pleasure to giving this talk both to my new community and part of an old community I used to belong to. Recognize some of you. I hear there's a big supply of young blood into the holography field. In fact, my PhD was actually in holography. And I was, in fact, my most cited papers are still in some early papers on entanglement and how to compute it with holography. Back when I was writing those papers as a grad student and even as a postdoc, no one cared. The field was kind of dead. Then I moved on, and so it's especially a pleasure that the Aspen Center for Physics has required you to come into this room and listen to me for the next hour. Right. Jokes aside, I kind of represent a workshop here that's extremely unified in asking one question, which is extracting computational power that's deeply embedded in the dynamics of physical systems. At the same time, the workshop is extremely fractured in that each physicist comes with a different physical system in mind. And to get at the big questions here, I'm going to have to use examples from particular systems that are familiar to me. But at the same time, I want to keep encouraging you to swap out particular physics that I show with other physical systems, quantum systems, optics, photonics, electrical systems that the diversity of people at my workshop are thinking about. So with that warning, let me start with a very concrete problem. How do you maintain the temperature of this room at a fixed number? Classic control theory problem 101. You open even an undergraduate textbook, they will draw a block diagram like this that represents a PID control loop. What it's really saying is measure the temperature of the room, subtract from it the set point you want. That error signal integrated over time multiplied by a gain parameter. That integrated error signal should be used to turn on the furnace or the air conditioner as the case may be. And if you do that, this will work really well. And what's great about control theory is they don't really care how you implement the sensor, how you do the integration, how you do the actuation, none of that matters. It's a general purpose solution. Here is
PID.
Great. What I just mentioned is the I, where you integrate the error over time to do it. Simpler method is just proportional feedback, where you just literally take the instantaneous value, do it. This D, which is derivative, the integral version works, has some especially special properties that are useful. What happened to my slides? Right. And I do welcome questions during this. Thanks, Simeon. Here is a completely different way to solve the same problem that might seem silly on the face of it. At least for 0 Celsius. Take a glass of water, put an ice cube in it. Temperature of this water is going to stay at 0 because if you put some heat into it, it'll shrink the ice cube, take heat out, it'll grow the ice cube. It's so familiar you may think it's trivial, but it's actually a profound property of first order phase transitions. If you remember from whenever you took such a class, or maybe you didn't, like I didn't, that's Maxwell's construction. Free energies have to be convex. It makes free energy functions non-analytic. That's what leads to this behavior. It's not specific to water. This is specific to first order phase transitions. Trivial or not, let's think for a moment about pros and cons of doing it in this way. The right-hand side, it's clearly the con, the downside, is it feels more specific. You need to have a first order phase transition. Given that problem, it's a huge set of other advantages. It's way simpler than the left-hand side. One thing, it's a passive system. There's no energy required to keep this going, unlike the left where you need to sense and compute and do all that. There are 3 separate actions on the left-hand side, sensing, integration, and actuation, secretly hidden in the dynamics and kinetics of this phase separation. Okay? Except there is no separate person or molecule playing those parts. It's all integrated in one. And as a consequence, it's often a much more compact solution, much more robust solution that's less to break, right? So those are some advantages. You may think it's an interesting curiosity, but as I mentioned, it's a passive system. The energy comes from the input itself. You may think it's a curiosity, but actually, there's a version of this that's keeping all of you alive right now. And many of your most of your cells often have to solve the problem not of temperature homeostasis, but concentration homeostasis. Certain molecules you want to hold fixed at a concentration, even though the environment is bothering it and trying to fluctuate it around. For a while, what people in systems biology did was look for molecular circuits inspired by a control theory textbook. They looked for a molecule that could sense, integrate, and then feedback. Sometimes they did find it, but one of the most convincing examples in recent times, Zechner's lab in Dresden then realized cells actually solve this in some cases with the ice cube and water analogy, except a different kind of phase transition called a liquid-liquid phase separation. Actually quite counterintuitive if you haven't heard of it. You just mix certain molecules in water, you make it homogeneous, and let go, you'll actually phase separate where there'll be a dilute phase with a stereotypical concentration and a dense droplet of the same molecules. They just want to hang out like that, dense and dilute. Those 2 concentrations are fixed because of a first order phase transition. If you dump more molecules in, the concentration will not rise, the droplet will grow. Remove molecules, the droplet will shrink. Same reason as the ice. And in fact, the fact that most molecules in the cell do this, or many molecules in the cell do this, has been one of the biggest discoveries in biology in the last 15 years. This paper showed that that helps buffer concentration fluctuations in the cell. Hard to see the data change the total amount of the molecule by a factor of 10, and the concentrations change by less than 10% because of this. At least in this case, biology seems to exploit, needs to solve a certain control problem, exploits a highly physically embedded solution. And really that tension or that choice sort of at the heart of this workshop that that I'm at. Okay. Broadly speaking, success of computer science over the last 70 years is completely avoiding the right-hand side. Is to have the separation of concerns, abstraction first approach. Why is that so successful? Well, it's extremely general purpose. Somebody writing software doesn't need to know what kind of hardware it's going to run on. But you do give up a lot by doing that. And so essentially this workshop we are thinking through what the benefits are of going more to that side, right? Or maybe finding some point right in midway. That said, why are we looking for those advantages? I mentioned advantages of energy and compactness, robustness. Why exactly? Broadly, I'd say there are 2 motivations. Oops. One of them, my slide comes live. One of them is something that you're all responsible for. Just like me, I'm sure you're sending off sometimes profound queries to ChatGPT every day. Each of those queries costs a lot of energy. Training ChatGPT costs energy, and every question you ask it costs energy again. Right? When my slide comes on, you'll see probably heard people are talking about building nuclear reactors to sort of power the next generation. And all of that neural network architecture to some extent is still built on the abstraction first approach. In fact, I would say the dominant motivation for this workshop is essentially asking, can we use some of those physical platforms listed there, do the same kind of computation in a more energy efficient way? It's a dominant motivation. But personally, I represent maybe a different community with a somewhat orthogonal motivation, and so I want to spend an extra minute on this because I think it's relatively mainstream thinking in biology these days over the last 25 years, but maybe not to physicists outside of the area. Which is computation isn't just about symbolic operations we do for human beings on a computer. It's also a metaphor that's useful to understand the behavior of how systems interact with their environments. In particular, in the biological context, people have found it useful to think about how cells interact with complex information coming from the environment. Here's an example. Certain strains of E. coli, certain strains of bacteria see highly structured environments where each time the temperature goes up, oxygen levels drop about 20 minutes later. This is because they often enter the mouth where the temperature goes up. There's still oxygen, but a little bit later they're going to predictably fall into your guts without oxygen. What this paper showed is when the environment is structured like that, after that period of training, if you take those bugs and raise the temperature on it, it starts expressing anaerobic enzymes even if there's oxygen present, as if it's anticipating that correlation. Then you take those bugs, put them in a fake lab environment where you anti-correlate it, right? Raise temperature, increase oxygen, and they forget that correlation and learn the new one. Okay? These phenomena exist. The question is how. How do they learn it? Well, they can't be doing backprop. They must be doing something else. And even forget the learning. How do they even just do that inference time thing? See something, say something. This is one-dimensional, but there are examples in higher dimensions. Higher dimensional inputs coming in, and based on some logic in that, they do something. And it's a puzzle because even when we understand something about the mechanisms by which they do it, it only deepens the puzzle. There is a famous example. It's called the BMP pathway. It's the reason you are here, because it's what helps pattern your cells. You're an embryo, and make sure one end is the head and the other end is not the tail, but the feet. And this pathway essentially looks at a 10-dimensional environment. There are 10 molecules in its environment, and based on this 10-dimensional input, it needs to essentially signal something downstream, like say on or off, like differentiate or not, based on some logic on this 10-dimensional input. Except if you look at the wiring diagram, it makes no sense from an engineering perspective. There are 10 sensors, 10 inputs, except the sensors are not diagonal. Each sensor binds multiple of these molecules. Then further, these sensors get in each other's way. In fact, the sensors as suggested here are all actually split sensors, each of which can hybridize with the other sensor and get in each other's way. And then there's further lateral interactions all the way down. And this wiring network itself is not completely reliable cell to cell. Yet, this is experimental data. Let's say look at a two-dimensional cross-section. The x and the y-axis are two of the inputs that you scan. The heat map is whether that particular cell type said activated or not activated. They do highly predictable computations that, from other reasons, we know they need to do for this embryo to develop properly. One of the cell types essentially seems to compute a ratio of these two, activate if the ratio is less than 1. One of them, something simple, just adds them up, activates. The other is what they call an imbalance detector, like an XOR gate, where it's active if one is in great excess of the other. All of this raises the question, how can something that's hot, wet, and sloppy, just molecules bumping into each other with somewhat unpredictable interactions, nevertheless achieve such computation? That's the question. Since, especially with these days with science under attack, I want to sort of underline this point. Yes, it's great to build things. There are many engineering challenges, but the natural world is fascinating in its own right. We don't understand a lot of how we are made in the first place. And of course, if you figure this out, principles behind this, in particular, it could help synthetic biology. These principles maybe will also tell us how to build other kinds of computational frameworks, but at least for me, the personal motivation is just how does stuff that already exists, how does it work? even if you don't want to exploit it for anything else. Okay, so that's roughly the motivation. Sometimes I also like to explain how this advances statistical physics. The theory of disordered systems is a field traditionally focused on random disorder, Sherrington-Kilpatrick, random matrices. But anytime you think of a system that's being trained for something, it is not random at the end of that training. It's a highly atypical member. What can you productively say about those kinds of systems? Pause here, because I'm going to get into some actual, examples of systems, but in case somebody has a question about the motivations here. Highly motivating? Everybody's on board? All right. So, so the example I want to walk you through is a familiar one, separating cats from dogs. And I want you to think of that problem in the following way. You have a neural network, and here is a space of all images. It's a high-dimensional space, as many dimensions as the number of pixels. What does it mean for a neural network to classify it right? It means you tune the parameters such that that neural network imposes a decision boundary in the space that separates cats from dogs. Neural network does it in a complex iterative manner, where there are many layers, it does all this computation in between. Doesn't matter. At the end of the day, it puts in a nonlinearity decision surface between cats and dogs. So if you want this same behavior out of a physical system, one option is to mimic neurons one at a time with physical elements and essentially mimic the mechanism through which neural networks do this. That's the abstraction first approach. What I want to walk you through is our attempt to instead ask, can phase boundaries, first order phase transitions in a phase diagram, effectively directly mimic decision boundaries? Here's the phase diagram of water. Maybe some of you know, if you've read Kurt Vonnegut's Cat's Cradle, ice actually has multiple crystal forms. That's the phase diagram. And you can think of that phase diagram as essentially a classifier of environments. Certain environments are mapped to one output, others to another. The outputs are different crystal forms of ice. That might seem like a stretch, and it's not a great idea. It's not a useful idea in the case of water, but it turns out to be useful if you think about what people call multi-component systems. Water has one component, H2O, and that's it. Many of it, but that's just one thing. But in complex materials, in biology, you have a large N number of species. We don't usually think about this in physics. We do field theories, whatever, you have a few fields. Well, actually, I guess in QCD, energy physicists have had the courage to pretend 3 is a large number and motivate studying many, many fields. But outside of that, you have a finite number, a small number of fields. Similar idea, if you have many kinds of molecules, turns out you'll have a phase diagram which is now high dimensional, because anything that affects the phase behavior of that complex molecular group is an axis of the phase diagram. And so now, if a cell or a material wants to deploy different responses to different environmental conditions, you don't need to build a molecular circuit. You just need to use parameters such that the phase boundaries separate conditions. Right? There's another big advantage to this, which is when you do things like on the left, the input and output are often symbolic. The neural network tells you, oh, it's a cat or a dog, then you might have to still use that information to go to the grocery store and buy milk or buy a bone or whatever, right? To do something with that information. In this case, though, the output is not symbolic. The output is, earlier was a crystal of ice, now it's a phase, a liquid phase, let's say in biology, which is often the functional response. It's the equivalent of the milk, the bone. So the output is what you wanted to do with that output information in the first place. So it's highly compact, has all the other benefits I mentioned. Like I mentioned, actually, this is actually biologically inspired because the idea of liquid-liquid phase separation has been one of the big discoveries in the last 15 years. But you could go further. Neural networks, if you don't like the decision boundary, there are ways to train it such that the decision boundary changes, right? You can train it on a computer. What we've been asking is, could the same thing happen to a molecular soup? Have a phase diagram that you don't like, you show it examples of what you'd like. Can there be just natural molecular processes in there that we call molecular Hebbian learning that changes something? interactions between molecules, which reshapes the phase diagram. So that's essentially the research program. Like I said, I'm talking about phase transitions. Pick anyone in the workshop, they'll have their own physics. Right? We heard some interesting talks about just scattering of waves being used to do these things. And broadly, most of us here have one of two kinds of questions in mind. One is, what are those learning processes that reshape them? And just as important, set aside the learning question, what about a physical system makes it a more or less powerful computational device? For example, if I found out that phase boundaries had to just be linear surfaces, that's sort of like saying, yeah, this is kind of a one-layer neural network. If phase boundaries turn out to be extremely flexible, then it's a more powerful system which can sort of respond based on higher order correlations in the environmental inputs. For neural networks, we have a theory already of like how depth affects this expressivity. Physical systems, we are still trying to figure it out. Right, so I'm just going to walk you through really one example from our work of how this can actually be successful. What we tried to ask was, we take a supermolecule that have no interactions and train them to have a phase diagram. You can't do this with water. Water is what it is, it just has a phase diagram, but if you turns out with this large N idea, you can actually do it. The inspiration came actually from Hopfield's 1982 paper, which was in a way the foundation for the Nobel Prize this year. There are many cool aspects of this paper. I'm just going to focus on two. He showed, take a set of neurons with no interactions right now. And imagine you impose certain states, firing states. Think of these as spins, you impose certain states on them. And during that imposition time, there's a learning rule that says neurons that fire together wire together, the classic Hebbian rule, which is extremely simple and plausible because it's just local. Two neurons fire together if they wire together. And you do that for multiple states, you end up with web. And he showed that that thing is not a mess, at least in some parameter regimes, has a energy landscape with stabilizes those states, and that can be used for a computation then. Sometimes autocomplete. You start with a bad image, it can fix it. Inspired by that, we didn't try to mimic it element by element, but we just said, imagine you take a supermolecule that currently has no interactions, then in a period of training, some physical forces, put them in some spatial geometries, okay? There are many kinds of molecules, just like there are many people here, and the training period, we make them sit in some arrangement like this, and we assume Hebbian-inspired simple molecular idea, which says molecules that hang out together grow to like each other. Right? Two of you sitting next to each other grow to like each other by the act of sitting here for a while. Except we train it with multiple crystalline phases going through this process. And then our question is, will that system have a phase diagram that's interesting? Okay. Inspired by Hopfield, but I just want to point out, molecules are not neurons, they're not linear threshold devices. All they do is diffuse in 3 dimensions, bump into each other. No notion of a molecule firing and that. What I could show you now is just assume this rule. I could go ahead and show you what cool stuff we could do with it. Really, the way we made an impact in this area was you show that rule to a chemist or a biochemist, they'd say, well, what do you mean? Molecules can't change who they like and who they don't like. Really, our impact here was driven by figuring out that there's actually a broad class of molecular processes, both in material science and in biology, which makes this thing happen. Yeah?
Do you mind explaining just a little more on the left-hand side, how you go from, like, what defines the energy landscape, and then how you go from that to auto-completing?
Thank you. So JIJ, just think of it as a spin model. These are a bunch of spins. JIJ is an interaction between two spins. The Hamiltonian in mind is just the usual JIJ, SI, The idea is you let this learning rule play out, gives you an interaction matrix that looks like that web. Just take that Hamiltonian and ask what are its ground states. What is its energy landscape overall? And you get that.
And go from that to the dog.
Oh, sorry, I should have explained. So you have this energy landscape. These minima correspond to the image of a dog, and I didn't show you what the others are. Maybe it's a cat, it's something else. And Hopfield's famous idea was this can be used for computation in an almost trivial way, which is through initial conditions. You initialize the neurons with a partial image of a dog, which means not at the minima, but here, you just roll downhill. The computation here is almost trivial. It's just attractors rolling downhill. And thank you for that question, because there'll be a contrast in how computation happens in this physical system. It's not a downhill process, but rather a saddle crossing process. It'll be controlled by saddles instead.
But in your setting, the JAJs depend on time.
Right. So, well, it depends on how you read Hopfield. So, I would say mechanistically, this is in some sense what Hopfield has in mind. Like, imagine a Hebbian rule playing out, and it depends on time. But as it's during that time, you experience these states. So, if you integrate this, you get the same equation you have in mind, a rank 3 matrix, right? From a computer science perspective, you just integrate it in time and just tell me what the Jij is. I'm saying there's a plausible neural mechanism that gives rise to that. Right? And especially for the molecular case, I want you to have that in mind. About this? It's okay? Because we're actually having in mind, just like for the brain, there is a dynamic process that sets the Jij. It's not instantaneously set. We realized actually that people already had molecular processes that do the other equation, which essentially says two concentrations of two species are high in the same place, same time, you grow to like each other. I usually go into this in quite amount of detail because I'm usually giving this talk to biochemists and chemists. I want to be a little more schematic here. Like I said, actually in response to Natalia's question, neurons that fire together, wire together. People love this rule because we know molecular mechanisms for this, how synapses wire. For molecules, how could that be? Essentially, our insight was interactions in question are not direct interactions. It could be effective interactions. Okay? Maybe Hakan and I don't like each other, but maybe we both like Florian. Then that's, if you integrate out Florian, then there's an effective interaction between Hakan and me. That's modulatable by the amount of Florian, because remember, these are molecules. Is present at 10 to the 10 or 10 to the 5, or depending on the size of the cell. Okay, it's that simple idea. I think you already got it, but I put together a slide to communicate it in a slightly more high energy-ish language. You have some coupling between fields. You can't change parameters like, let's say, an alpha. Some number in the Lagrangian, you can't change it at some energy scale. Coupling between phi I and phi j, there's a product with another field, G. G is not a number, it's a field. And if you effectively integrate out that G, you get an effective interaction. This part you don't usually think about in high energy physics, but it's relevant here. Effective interaction depends on what that G is. And G, if it's changing slowly enough, G responds to phi I and phi j, and hence can depend on the history of what phi I and phi j did. The G can depend on whether Hakan and I spend time together or not. Essentially, yeah.
So we know each other for a while now. I just wondered, you've presented one part of the, how this kind of Hopfield network works once it's trained. Right? You said I started with a partial image, and it goes down and on. But with training, how does that work? How does that work in practice?
The real brain?
I guess these parameters self-adjust while I'm showing some examples.
It's a great question. So traditionally the way people, the left-hand side is not the real brain, it's all models. The way people think about it in the models, it's a period of training. This is why Natalia's question also came up. It's a period of training that sets the Jij, and then you freeze it, and then you do the inference. Separate those two. The experiment I'm going to show you, that's essentially what we did as well. But at the end of the talk, I will mention future direction we are doing. Actually, that's come up in our workshop too, the sleep-wake-like methods, where training can keep on going, because the sleep-wake methods do not overtrain. It can be ongoing, just it's happening at a slower time scale. It's easier to think.
Okay, so that's plasticity.
Exactly. But for now, to simplify, just imagine there's a period of training, and then there is the inference time where parameters don't change. Right. So usually for biologists and chemists, I use this language instead. There's some interaction matrix, large one, disordered. You freeze that, but then if you integrate out the blue stuff, you get an effective matrix for the visible species, which is not a constant, even if the big matrix is a constant, because the little one depends on concentrations of the hidden species, and that can change. And in fact, what we realized was Other people over the last 10, 15 years have found many, many examples of processes that give you this molecular Hebbian rule, both in materials but also in actual biology. And so that let us essentially do an experiment. We need a system with many, many kinds of molecules. We could do it with proteins. It's easier to do it with DNA, where each sequence of DNA counts as a different species. What we did is what I just described. This simple molecules that initially have no interactions, pretended to hold it together in one crystalline phase that resembles an H. It's a finite crystal. And then in an A, and then in an M. These are the three phases we want these molecules to form. And while they are held in that phase, they are learning these glues. The glues are being created that make people like each other. Okay? Many others have actually done this learning reaction in the lab. In this paper, actually, we simulated that and then simulated those experiments of other people and did just put those glues into the test tube. At the end, we have this. We asked, what is the phase diagram of this soup? First, simple experiment. Put all these molecular components together. Essentially a Hopfield-like experiment, but with a special pattern. And think of that special pattern, all equal concentrations as one special point in the phase diagram. And we found exactly 3 crystalline phases, the 3 it had seen during training, nothing else. But that same test tube, if you now go to a different part of this phase diagram where some molecules are at a higher concentration, you get only the H crystal. Think of this as water freezing into multiple ice crystals. In other parts of this phase diagram, we get specific crystals. In fact, you can go poking around and you find whole regions. You get one of these 3 phases. You can quantify it because we could put fluorescence traces that are specific to which crystals are being created. But the summary is this really simple, especially if you're a chemist, a really simple process that creates glues based on people hanging out together, lets a soup of molecules assemble multiple phases at the same time. From what I said, it may not be obvious, but there's actually a pretty non-trivial property. For those of you who know about Hopfield memory, this might ring a bell. In theory, we asked what would happen if you kept trying to learn phases this way. And it turns out, as you learn it, the system is getting more and more promiscuous. There are problems that can happen. The 2-dimensional or 3-dimensional nature of these assemblies has a certain error correcting property that prevents problems from happening until a sharp threshold, which is again a phase transition, but now the number of memories or the number of phases you've tried to learn. That capacity, before this thing becomes a glassy system, depends on the dimensionality, the structures because of an error correcting property, and the number of species. Partly bring this up because it's both reminiscent of Hopfield associative memory. It's also not, because for one thing, with local interactions, Hopfield neural networks have no capacity at all. And this kind of physical nature of error correction doesn't arise in that. The capacity is a different thing. But in fact, this is related to Fatih Dinch here. I don't want to call him out. Oh yeah, he gave a nice talk at the Blackboard yesterday and mentioned continuous attractors in neuroscience. And this, the math of this turns out to be related more to that.
Sorry, why the capacity is different from the usual capacity, memory capacity of the
Question, I would say, why would you expect it to be similar in any way? Because the system has, in a way, nothing to do with the real thing, because these are molecules diffusing in space, bumping into each other, and aggregating, sometimes not aggregating. So for one way to say it is, first of all, this is only local interactions. Hopfield model is all-to-all interactions. I would say I don't even know where to begin, because neurons are connected to each other. All they're doing is influencing each other's state. If you just think through a little more, there's no way to really map states of a Hopfield model to molecules diffusing in 3 dimensions, sometimes bumping into each other.
you only take into account the local interaction.
I would say,
See where, how it comes into.
so I showed you experiments. Experiments are just molecules dissolved. The theory did no neural network theory. We just analyzed diffusing molecules in a 3-dimensional system. Right? I understand the spirit of your question, and it's partly because of the slick nature of my slides. It sounds like that. But the real question is, why would you expect any similarity today?
I'll just clarify a little bit more how the training works. So if I understood what you said, so the training is done in silico. Is the idea that, I take it these are DNA tiles of some kind, and so if you just assemble them in the computer to make the age, and look at what the interface histogram is, this then trains you what the concentration of the corresponding groups ought to be.
Yes.
So we could gain that.
Yes.
So, like, if you were to actually try to do the training in vitro,
Yes.
you have to artificially hold those tiles together, or how would that work?
Exactly. So during the training period, so this is crystalline assembly. Later I might show you liquid assembly. But during training period, these molecules are templated some way, either in space or in the liquid case, it can be well mixed. It doesn't matter. But there are spatiotemporal correlations in concentration. The experiments I said others have done essentially creates glues enzymatically. The two things hang out together. This is actually the principle behind DNA microscopy.
But like in this version, you would do something like, so you sterically hold the H together, then you flush in the different kinds of interface glues,
That's one way.
and then you pull out the H and dissolve it back out and measure the something like that.
That could be one way. And actually, there are one of the experiments I mentioned. That's the death of the lonely glues idea. There are the idea on top creates things. One at the bottom is also more biologically it happens where I won't get into it, but RNA essentially dissolves when it's unstable when it's single-stranded, but saved when it's double-stranded. There are other mechanisms as well. So chromatin, parts of a polymer that hang out together grow to like each other because of reader-writer mechanisms. And then there's the other case, two transcription factors together express a scaffold molecule that likes to bind these two together. But yes, your question is in particular, this work is about crystal assembly, so the training time is about spatial structure. What we're doing now, it's liquid stuff, so you just mix it in.
I also have a question related to Natalia's question. So first, what do you mean by dimension of the pattern? And the second is, I would also know whether that depends on how many genes you can actually bind, or you can actually have for one molecule. I didn't see that at all in the formula.
Great questions. Actually, both of those numbers are the same thing. So there are two concepts. So the dimensionality, I mean, so we have a test tube with molecules dissolved. But when those molecules, when you cool it, molecules come together and assemble a structure. That structure could be two-dimensional or three-dimensional. That's a dimensionality, I mean. And the answer to your second question is in 2 dimensions, each molecule at a given time can only bind, let's say, 4 glues. But number of glues in the system itself is larger because just like in a feel-like system, like each molecule in principle has many, many binding partners. It's just when recovery happens, they're bound to exactly 4. That is what leads to the error correction. So that was about training something. At the end of it, just getting some phases in some parts of a phase diagram. An interesting computational question is how flexible are these phase boundaries? Right? Can I wiggle them any way I want, or is it just linear surfaces? One way to get at this is to play a adversarial game. I pick on Logan. Pick somebody who is not not friends, and play an adversarial game. We are friends. It's just let's say we like to challenge each other. Picks 3 points in this phase diagram, labels them pink, and 3 others labels them green, and challenges me to put a phase boundary that separates them. And he doesn't make my life easy because he interleaves these points. If I succeed, then he can up the ante and pick 4 points, 6, 8, till I fail. The point at which I fail, because these are not infinitely flexible phase boundaries, tells me how flexible our first order phase transitions in say liquid to crystalline assemblies. So actually in the paper we did this kind of empirically, where instead of asking Logan, we just downloaded grayscale images which have as many pixels as we have molecules, so we could treat each image as a concentration pattern, and just group them arbitrarily. Dorothy Hodgkin, Horst, and Hopfield, these patterns have nothing to do with each other. We said those should be one phase, the other should be the other phase. And actually in this case, what we did was not the Hebbian, we already had this Hebbian trained system. We just trained it in a reservoir mode, but the only choice we get to make is which pixel position corresponds to which molecule's concentration. It's a choice you make once and for all. Just looking over those choices, essentially how the input goes into the system, can you solve it? And making the problem more and more difficult until it fails. So First, with some small set, things worked, meaning we trained it with this set. Then you see Hodgkin, Hopfield, or a horse. Only the red trace quenches, meaning one crystal phase assembled. Avogadro, Abbott, and Anchovy, different phase. And then with the next. Especially with this audience, do you recognize all the scientists on this? The first column are scientists famously associated with crystallization and say physical chemists. The second column are computational neuroscientists, real neuroscientists. Moser won the Nobel Prize for these continuous attractors. Space Hopfield, physicist, who won it this year. In fact, Larry Abbott and the animals are the only ones who haven't won a Nobel Prize on this slide. Hopefully it bodes well for him. All right, so that slide worked, but then the whole point was to keep making it more and more difficult until you see failures, and it gives you some sense of how expressive this is. But actually, let's see, I'm running out of time, so let me just wrap this up. I'm showing you some theory we did actually well in advance of these experiments that really get at how physics decides how expressive the system is. I know most of you here don't ever think about molecules or thermodynamics for that matter. I want you to just get a schematic sense of how computation could be enabled. So somebody, I forget, maybe Natalia asked me, in Hopfield model, computation happens through attractors. It just roll downhill based on initial conditions. This system, after training, there are 3 deep attractors corresponding to the 3 phases. That's what it means to have thermodynamic phases. But initial conditions, solvated molecules in a test tube, start at a metastable state. Why is it metastable? Because even if molecules, like you dissolve salt, water, before it can form a salt crystal, it has to nucleate the salt crystal and then form it. Just how physics works, I guess. But the point is there are saddles. Real computational power is the system starts in the metastable state and is trying to figure out which saddles are easiest to climb over. That's what decides where it goes. Why is that helpful? It turns out each saddle is associated with a monomial, which is a product of some of the input variables, in our case, concentrations. Why is that? Because each saddle, nucleation theory, is a partially formed critical seed, the hardest thing to form before you fall over. And it's a product of concentrations in that seed. And so really how the system decides whether to go to the green output or the red output is it adds up. It's a huge number of saddles. It's many, many molecules, high-dimensional space. It adds up monomial corresponding to all the saddles that go to the green structure and all the saddles that go to the red, and subtract it and ask, is this positive or negative? Okay? Intuitively, the system wants to go over the easier-to-climb saddles. The product tells you how high the saddles are. Then you immediately have an understanding how expressive the system is, because it's just the order of that polynomial tells you how flexible this boundary can be. What sets the order of that polynomial? I did not take thermodynamics or StatMech because I was trained as a string theorist. I thought, who would ever need that to make a living? So I'm not going to pretend this is obvious, but what I'm saying is textbook material for, let's say, even a chemist, right? College freshman courses, they learned this. They learned what sets the order of this polynomial, which is essentially temperature, if you wish. If the temperature is low, it's easy to nucleate. The critical seed will be really small, maybe just one component. Monomials will be linear. The whole polynomial will just be a linear polynomial, which is not very interesting. The system, all it can do is ask a weighted linear combination of concentrations and ask, is that weighted some better or this weighted some better? Weighted average. But the temperature is high. It's really hard to climb. Saddles are far away from the metastable state. Big structures, high order polynomials. You get high expressivity. Essentially, it's asking high order correlation questions about the input for making a decision. There's a problem, which is if you're at high temperatures, it's hard to nucleate. The system is going to go back and forth, keep trying to climb the saddle, fall back many, many times before it finally goes over. So from that, you can derive sort of relationship between how long it takes to make a decision, the expressivity. What order correlation functions can this compute? And it turns out that same relationship has been seen in like these sort of stochastic algorithms for solving satisfiability problems like Sudoku or crossword puzzles. Somewhat similar reasons, because those algorithms like to backtrack and try things out. So for complex problems, you really want to keep backtracking before you do it. So here it's from temperature, but that's really meant to be an invitation to think about the expressivity of systems. I know Hakan, for example, thinking about the expressivity of quantum systems. Yesterday, or was it 2 days ago, Clara gave a great talk on the expressivity of wave scattering. Sounds like a linear system, but turns out it's not. So really, collectively, we are trying to ask, what about the physics of a system makes it more or less complex this way? Oh, and maybe just one last slide on this part, which is, I started the talk by promising that physics first has some benefits. In this particular problem we solved, many people have tried this before, molecular neural networks, often with the same kinds of molecules, but by mimicking neurons element by element. Okay? So just to summarize some advantages we got as a consequence. The problem we solved was about 6 times bigger than the state of the art, and we used way fewer molecules per input dimension than the state of the art. In fact, I would say we used the smallest number possible because the number of molecules is the number of input dimensions. Hard to imagine being below that. It's also more expressive. It's a bit of a technical point, but I think it's an important one. In this field, people use what's called purified components, whereas we used unpurified molecules, which means when you have a test tube and I say, oh, I have 50 micromolar of some molecule, actually nanomolar in our case, 50 nanomolar of some molecule, more than half of it is not actually that molecule. It's broken variants of that molecule. If you pay a lot more money, you can sort of purify it and get only the perfect ones. We did not. We think it still worked because it's a first order phase transition. It's a pretty robust thing with fluctuations in exactly who binds how. Right. How many minutes do I have? 7 minutes. Okay, so I'm going to wrap this up quickly. What I've shown you so far is just systems trained in the simple Hebbian way. I mentioned to Hideo, we did that training part, simulated and did it. Others have done this, that learning part for other purposes many times. But it turns out you can do much better if you alternate these kind of Hebbian processes, which sort of encourage a system to show a behavior, with anti-Hebbian behavior. And we discovered this accidentally many years ago with a mechanical system. We're trying to, this is a theory paper, train a mechanical pattern recognizer or fingerprint recognizer. And so let's say Florian here was pressing his fingerprint against a mechanical device and we wanted to unlock them. If it unlocks, then we do Hebbian learning to encourage that behavior. We don't want my fingerprint to open it, so when I apply my fingerprint and it does open, do anti-Hebbian, we stiffen things in this mechanical system. That to work much better. It actually turns out that this idea is a classic one, a much broader one in machine learning. People realized a while ago that in addition to Hebbian, you just regularize by sometimes making parameters go the other way. You can fix mistakes you did in Hebbian. You can prevent overtraining. Somebody asked about ongoing learning. You can essentially have an ongoing learning method. And I'll highlight one of them called sleep-wake training. So the other person who won the Nobel Prize this year, Jeff Hinton, was inspired by many of John Hopfield's papers. And he came up very early on this idea of sleep-wake to train what are called Boltzmann machines. And the idea is this. It's a phase called wake phase where you do exactly what I've shown you. You impose a behavior you want on a neural network, do Hebbian learning. Make sense? But then there's a sleep phase where you don't impose anything, let the system show its spontaneous behavior, do anti-Hebbian learning. Really, you need to compute these two things, and then update the weights based on the difference. Okay? That's been exciting because the whole rule is local, and you can prove various things about how much better this idea is. So lots of people, for example, Eric Winfrey, a while ago, proposed, wouldn't it be nice to have this in a physical system, like a molecular system? The main challenge is that this rule is local in space, but it's not local in time. There are these two phases. You need to experience them and then somehow compare them and then update. Okay? That has not stopped Sam Dillevu here, who in Andrea Liu and Doug Durian's group, who I think will be here next week, at Penn, built a completely autonomous distributed learning resistor network that no GPUs, just sort of trains itself as it sees electrical inputs to give the right output. But he solved this non-local problem by essentially having two copies of the system, one of which is experiencing wake and the other is experiencing sleep, so you can compare and then do it. Right? That idea may not be available to other physical systems, say biology or a material science thing. You have two copies. The question is, what can you do? So I want to leave you with an ad for, it's an ad? Yes, it's an ad. For one paper of ours that just came out, which again goes back to this physically embedded idea. Instead of starting with a machine learning idea, we just modified what physical systems, thought about what physical systems naturally do. Here's our proposal. It applies to any physical system, including neural networks, let's say. Usual Hebbian learning says neurons that fire together wire together. A simple modification is to say, well, actually, neurons that start firing together should wire together. But if they keep firing together, well, just forget it. Stop updating. Spend some time with somebody, two molecules hang out together, Hakan and me, start growing to like each other. After a while, it just tapers out. It's we haven't gotten to that stage yet. But technically, this is actually an idea that's enormously explored in biology because all your senses, almost everything in biology is of this type, doesn't keep responding. Your eyes, the only reason you can see both what's outside and your eyes can work in this room is essentially this idea of baseline subtraction. Something has been constant for a while. You just subtract out that mean and only respond to changes about the long-term mean. Okay? So our proposal is very simple. Just do Hebbian updates. Just grow tired of stuff after a while and stop doing it. Technically, how is that done? The simplest way is essentially integral feedback where like that synaptic current on which you're doing Hebbian learning, imagine there's another force that's trying to not let it change. So when two neurons start firing together, it looks like a step function, but really the signal goes back down. Okay. Why am I saying all this? Turns out that simple idea that has been explored for 20, 25 years in biology is secretly a time derivative operator. See that here. The derivative of a step function is a little blip. Essentially, it's saying update based on time derivatives. We've essentially simplified the system itself. The system itself doesn't always in one mode. It doesn't know about sleep and wake, but we demand a little more of the environment. We say the environment, lets you sleep. When it wakes you up, it should wake you up fast. Meaning when it's imposing what it wants on you, do that fast. That's the upswing of the sawtooth. And then it should let you go back to sleep. But the way it relieves that constraint should be slow. So there's an asymmetry in that time. When you pass it through this time derivative operator, you can show that that essentially does the same as the sleep-wake rule. What's cool about this is one thing, as I said, the system doesn't really know what's going on. It's just sitting in one state. But what's also cool, it's all, again, the sort of everything happening in one step. You're not having to compute the weight update, store it somewhere, subtract, and then update. All of that happens in one physical operation. I think actually Florian has a proposal of this type as well, where updates to the parameters of a physical system is just all of it. The computation, the updates, just happen in one swoop. Okay? And anytime you find something like that, it's more likely to work in a real system. So I'll end up by saying where we're now. I'm obviously most excited about this in the biological context. I forgot to fully edit this slide, but we're especially excited about this because we think the ingredients needed in that proposal. In the paper, we explain how this idea can work for resistor networks, whatever. I don't think we did optics, but resistors, mechanics, and so on. But the biological context, we think the ingredients needed already exist, completely uncontroversial. Just people haven't thought of those three ingredients working together. Here are the three ingredients that we're trying to put together. What is the system being trained? It's the molecular network that's used by your cells to respond to combinations of hormones. Okay, your cells experience hormones from other parts of the body. The way they compute what to do is by one of these messy binding interactions, kind of like the experiments I showed you. That's the system in question. For training, it turns out the rules I said on the previous slide, all it amounts to says some of the components in this pathway, if you have a bunch of them, if you have more of one component, it should make more of itself. Okay? Or rather, actually, more precisely, if some component's concentration is going up, in what's called the free phase or the monomer phase, it should go make more of itself. Extremely generic in biology. A molecule that leads to the expression of more of itself. Then we make a demand of the environment, which is that the hormones in question show up dramatically on a fast time scale and then go away on a slow time scale. Okay? We could debate how realistic it is, but at least we have simplified the demands on the biological system itself. With those ingredients, what we can show now in theory, but hopefully shortly in experiments, is imagine initially the two axes are the two hormone levels, and imagine a given cell activates just in those regions, but you want to actually change it to that input-output map instead. Just by having that cell experience those x's, each of them correspond to some correlated spikes of these molecules. After a period of training, you'll end up with the desired computation. What's really cool about this method, which is not really our idea, it's Hinton's old idea, it addresses the problem that Hakan brought up, is we don't need to turn off this training. This training can just keep running forever, because I won't have time to explain this, but essentially this training rule, if you keep letting it go, it'll stop doing anything when the sleep behavior looks like the wake behavior. That's when the spontaneous behavior of the system looks like the imposed behavior, the system is trained, this learning rule just stops doing anything, which is great. And if the environment changes, it's going to update itself and move on. I see Hakan get up, so I will summarize, actually my last slide. Basic take-home I would like to suggest is if you want some computation of molecules, photons, whatever, it's best if you let them do what they naturally want to do. What that means in practice is not necessarily take a machine learning idea or a computer science idea, ask how do you implement it in the physical dynamics of something. We really try to go the other way. See what they do, and then ask, well, can it accomplish some task that you care about? So that's not just my perspective, that's really, last few days we've had 4 talks, and I feel like this idea was sort of permeating all of it. Logan, I think, was explicitly saying this. That I'll stop, and just, I do have to point out who actually did this work. Much of what I talked to you about is more on the in vitro molecular learning aspect. Started with Zorana Zdravchik 10 years ago, and the latest updates were with Eric Winfrey. Synthetic biology directions, really with Michael Elowitz at Caltech. We did, along the way, early on, some things on mechanics of the same type by a grad student then, Nahi Stern, who's now faculty at Imolf. And then the future will be some cell biology versions.
Floor is open for some questions. What I'm going to do is I will pass the microphone to anyone who wants to speak, ask a question.
Exhausted you folks. Oh.
So my very first-grained understanding of biology is that everything is a Hopfield network. And when you were showing this example with the molecules, I was thinking about how, for example, stem cells need to decide how to differentiate into, am I a liver cell, am I a skin cell, or whatever. So since you have this word cellular biology here on the slide, I want to ask, can you do this? Can you control which state cells differentiate into in the same way?
It's a great question. So that analogy has actually been made by people in the field as well. And I would say that one is more like matches Natalia's intuition. Cell state is decided by a bunch of interacting elements, but not really in physical space. It's gene regulatory interactions. So in some way, the models of gene regulation are really just neural network models, are variables with nonlinearities. You write the equations, Natalia is going to say you've just written down a neural network. On the other hand, not that you care about the learning, but the learning part doesn't apply to cell states. It's programmed in by evolution. But otherwise, there are people who are literally asking, how do you take a stem cell from here, that state, and not to this state? What is that control knob I should turn? By making the analogies to Hopfield. The Hopfield version I showed, because it's literally molecules in 3D space, don't immediately follow from the hot field analogy, I would say. I'm also not sure I would agree with you that much of biology is just hot field repeated many times over, but yes. They also had his hand up.
It's great talk, thank you. I wonder if it's biologically plausible to extend these ideas to represent dense associative memory
Great
with
question.
improved capacity,
Yeah.
dramatically improved capacity, etc.
Great question. As you probably know, these dense associative memories, I think in one of the follow-up papers, there have been proposals for mechanistically, how do you get these? So for those who don't know, the original hot field model is pairwise couplings. These dense associative memories have much better performance, but you need higher order couplings. Physically, it's hard to imagine direct higher order couplings. They could emerge again from integrating out something. The molecular realm, we haven't figured something out. In the neuroscience one, I've seen people have proposals of these astrocytes, which link many neurons together, and if you integrate those out, you get effective. But I'm not really a neuroscientist. You should ask Fatih. So. Hideo had a question.
Question in the back. You could ask the microphone.
Yeah, thanks. Just going back to this thing with the pictures of the scientists and the HA&M. So in this initial round of when you're just trying to train the 3 phases of HA&M, so the question would be, do you have a sense of how many glue species you need to accomplish this 3-phase diagram? Because then, I want to try to think of what you show with the images as being this usual story that get nonlinear classification, one trick you can use is just dimensional lifting.
It's what?
It's dimensional lifting.
It's dimensional lifting.
Like a nonlinear dimensional lifting will make things that are nonlinear in some lower embedding. They will generically become linear in the higher space.
Good question. I haven't thought through this. The concrete question, how many glues? I don't have the number off the top of my head, but essentially each assembly, what is it? It's roughly proportional to the area of the structure, because each person roughly gets 4. You just multiply that by the number of memories, so it's linear in that. I'm not sure. So if you train more and more memories, there are more and more gluing species. I'm not sure if that I'm not sure that those gluing species in one version of this in our paper, they're not actually interfacing with the external world at all. They're just background fields. You don't see them. They just happen to be in the tube. You only interact with the subset. It's not clear to me it's playing the role of that dimensional lifting. I would say in the end, fundamentally, our nonlinearity is coming from this being a many-body effect, phase transitions, because it's a large crystal. I'm not completely confident because I haven't thought through it.
I had a question.
So, Arvind, I wonder whether one can expand this idea. Not very much into this previously. To think about compositionality.
Great question. So, I think first I want to state something, a philosophical difference. So the question is about compositionality. So in computer science, there's this correct idea that computational paradigms are most powerful if you can compose computation. That is, you have an input, it comes out, some operation. And if that can be fed into, that's what you mean by composition, right, into the next one. For example, this is how deep networks work. And there was just a discussion at a Blackboard about this. That's true. And it's especially true if you're trying to build general purpose computers, which is maybe the focus of this workshop. I would argue that in the biological realm, evolution has a problem to solve. It just solves it. It's not trying to build modules that can then be reused, except in some cases. I'm personally biased against looking for compositional solutions. That said, this particular version does have a compositional aspect. Experiments we did, these were 2-dimensional crystals. So the computation happens in nucleation and assembly of a 2-dimensional crystal. So that 2-dimensional crystal then can grow in the 3rd dimension. And there's a second, and the output of the first computation is its 2-dimensional layer, which then influences nucleation, a different kind of nucleation, what's called heterogeneous nucleation on the pre-assembled surface, which then does, right? In fact, as you know, Eric Winfrey's whole theories are about self-assembly can do computation, and this stuff can be composed if I must, but I would rather not. Thank you.
Actual science fiction.
Things are wrong.